Hello, I am Seth Richard, and I am a lab manager here at the Hormel Institute. I was actually raised here in Austin and graduated from Austin High School before getting my undergraduate degree in biology over at the University of Minnesota, Duluth. So I'm going to take a few moments to explain some of the research that we do here at the Institute. So our section name is Cancer Cell Biology and Translational Research, where we do a lot of basic research or a lot of in vitro work where we compare normal and tumoral cells to try and understand the differences between them. So we're hoping to identify targets that will help us to kill cancer cells, reduce their growth, or even turn them back into a more normal phenotype. And once we do have a good candidate, we can move on to preclinical studies, which use animal models, which are typically mice and rats. And finally, if everything is very promising, then clinicians may take the lead and set up some clinical trials. So our lab here focuses on this earlier part, and I'll talk about one of our main projects we have in the lab that relates to the role of primary cilia in tumor biology. So first, a little bit about the liver. The liver is one of the key organs to maintain the homeostasis of our bodies, as one of its main functions is the production and secretion of bile. And this bile is essential for a lot of processes such as digestion and the absorption of food, and the elimination of some waste such as endo and xenobiotics. So bile is elaborated inside the liver, transported through the bile ducts, and into the gastrointestinal tract. So this is a 3D reconstruction of part of a rat liver. The brown cells are the hepatocytes, and inside that yellow is the biliary tree, a large network of small tubules that collect and modify the bile. Going on is a electromicrograph of the liver where we can see the portal vein, hepatic artery, and bile duct. And if we zoom in on the bile duct, you can see these little hair-like structures, which are the primary cilia, protruding from the apical surface into the lumen. So you can imagine bile flowing through here. So cilia are positioned to interact with the bile. Therefore, they're exposed to a lot of dynamic changes in bile flow, composition, and tonicity. But these changes are detected then by the cilia, and information is transmitted into the cell. So the cilia is actually getting a lot of importance lately, as it is considered the cellular antenna. But our lab, we wanted to start exploring the potential role of primary cilia in cancer using cholangiocarcinoma or bile duct cancer as a model. So we started looking at cilia in tumor samples and analyzed the expression of cilia in cholangiocarcinoma, and we found that primary cilia are significantly decreased, both in vivo, as shown in this normal and cholangiocarcinoma human samples, where the cholangiocytes are standing green and cilia in red, but also in vitro when we compare normal tumor cells in a plastic dish. Therefore, based on these observations, we're trying to understand what are the consequences of ciliary loss, why are cilia decreased in cholangiocarcinoma, and if cilia could be restored in tumor cells as a therapeutic approach. To assess the consequences of ciliary loss, we are using normal cholangiocyte cell lines and experimentally eliminating the cilia, and we found that the elimination of cilia transform a normal cell into a more malignant one. So to understand the mechanisms of ciliary loss in cholangiocarcinoma, we put our attention on molecules that are involved in cilia biogenesis, which pointed towards one protein, histone deacetylase 6, or HDAC6. So the structure of cilia depends on the stability of the microtubules. In this cartoon, the red represent the microtubules, and HDAC6 can modify the chemical structure of them, inducing disassembly. So, of course, we went and wanted to look at the expression levels of HDAC6 in tumor samples compared to normal, and we found that HDAC6 is overexpressed in cholangiocarcinoma. Therefore, we can imagine that in tumor cells, HDAC6 is much like a big wrecking ball that destroys this cellular antenna. and the loss of cilia helps with the malignant transformation. So based on these findings, we can say that we have a potential target to tackle our third aim there for the cilia restoration as a potential therapeutic. So we are using these cholangiocarcinoma cell lines and inducing the restoration of primary cilia by targeting HDAC6 with a specific pharmacological inhibitor. This one is tubistatin A, which induces the expression of cilia, which are shown here in red. Finally, we assessed the effect of tubistatin A in an animal model, or rats that have a cholangiocarcinoma tumor in the liver. And we found that the treatment with these HDAC6 inhibitors reduced the tumor growth. So again, this correlates with the appearance of cilia in the tumor cells. Therefore, our data suggests that the restoration of primary cilia is a potential therapeutic approach for this tumor. Overall, our data suggests that the restoration of primary cilia is a potential approach for cancer in general. And it's very exciting since a lot of other types of common cancers have been observed to have a lower cilia expression, such as not only cholangiocarcinoma, but pancreas, breast cancer, ovarian cancer, just to say a few. So, thank you very much for your time. If you have any questions or want to explore a career in science, feel free to contact us. And thank you very much.